To bring back a woolly mammoth, scientists have a massive living advantage. They can rely on the reproductive system of a closely related surrogate, the Asian elephant. Avian de-extinction functions under a different set of constraints. Unlike mammals, birds develop in a self-contained environment, physically bound by the walls of an egg. Look at the towering skeletal mount of the South Island giant moa. Colossal Biosciences has targeted this extinct New Zealand native as a prime candidate for resurrection. The immediate hurdle is sheer volume. This diagram compares a standard chicken egg to an emu egg, and finally to the egg of a giant moa. A fully developed moa egg was roughly 80 times the volume of a standard chicken egg. Even the moa's closest living relative, the emu, is physically incapable of laying or incubating an egg anywhere near that massive size. Genetic editing alone won't solve this. To resurrect a giant bird, science must engineer an entirely new incubation system capable of supporting embryos far larger than those of any living surrogate. Scientists have spent 4 decades attempting to culture bird embryos outside their natural shells, beginning with primitive laboratory tests in the 1980s. These early methods often relied on makeshift containers, from rigid plastic cups to suspended plastic wrap. Those early plastics had terrible gas diffusion. To keep the embryos from suffocating, researchers had to pump in highly pressurized, pure oxygen. This triggered a destructive cycle known as the oxygen toxicity loop. The concentrated oxygen generated free radicals that damaged the developing embryo's DNA, causing widespread developmental failure. Colossal's solution to this problem is entirely mechanical. This is their newly developed incubation vessel. It uses a 3D printed geometric lattice cup to provide rigid structural support, suspending a vanishingly thin bioengineered inner membrane. By redesigning the physical architecture of the container, they bypass the need for pure oxygen, dismantling a long-standing mechanical barrier in embryology. Colossal's membrane is manufactured to a thickness of just 20 micrometers. As this diagram shows, that precise thinness allows it to mimic a natural shell, permitting 21% ambient oxygen to diffuse in and carbon dioxide to flow out, all while trapping the necessary fluid inside. There is one mechanical limitation. A synthetic cup cannot provide the calcium carbonate normally scraped from the inside of a real shell. To compensate, researchers must manually add ground biological calcium into the fluid mixture so the chick can ossify its skeleton. With that manual adjustment, the hardware works. Colossal recently announced the successful hatching of 26 healthy chickens from these synthetic essels. As a pure engineering challenge, they have triumphed. They have successfully replaced the avian eggshell. But scaling up the hardware, the physical container, doesn't automatically solve the biological limits of the contents inside. We have to look at the wetware, specifically the yolk. An avian egg yolk is not a liquid soup. It is a single, incredibly massive cell protected by a highly delicate outer lipid boundary called the vitelline membrane. Colossal has hypothesized they could merge dozens of emu yolks together inside the synthetic cup to feed a giant moa embryo. But as the cellular animation demonstrates, biophysically you cannot simply press 50 massive delicate cells together without rupturing their outer membranes and causing the entire structure to collapse. You see the same profound biological size mismatch in the dodo project. The starting point for that effort is the dodo's closest living relative, the Nicobar pigeon, a bird that weighs roughly 1 pound. Look at the sheer bulk of the dodo's skeletal frame. A 1-pound pigeon is biologically incapable of producing the massive, naturally sized yolk required to nourish a 40-pound flightless bird. You can use a 3D printer to scale a plastic cup to the size of a football, but you cannot simply brute force the cellular biology of the largest single cell in the animal kingdom. Because ancient DNA is highly degraded, these projects aren't resurrecting true, authentic moas or dodos. They are using gene editing to create highly engineered transgenic proxy hybrids. Breeding highly modified, sentient creatures into completely artificial environments carries unknown health outcomes, raising serious animal welfare concerns. There is also deep opposition from Maori communities in New Zealand, who view the genetic commodification of their native species as a direct violation of their sacred cultural heritage. If we strip away the de-extinction marketing, this technology has a much more urgent application. For critically endangered species like the kakapo parrot, this artificial egg could be used to rescue damaged shells and bypass the severe infertility caused by inbreeding. It also offers a platform for the biopharmaceutical industry. With shell-free access to growing embryos, researchers could scale up the production of vaccines and therapeutics using transgenic chickens. The artificial egg may never raise the dead, but by saving living populations and streamlining medicine, the technology establishes its importance as a foundational tool for modern biology.